Sterile technique and culture maintenance. We live in a microbial world. Microbes are everywhere, on us, inside us, and around us. Microbes are much more important than we will ever be. The microbial world is the foundation upon which all life rests. For example, the mitochondria that make the energy in our cells and the chloroplasts that power photosynthesis in plants got their starts as free-living bacteria. Oceanic cyanobacteria, less than 1,000 of a millimeter wide, produce much of the oxygen we breathe today. Microbes in the soil nourish the roots of plants and transform inert nitrogen gas from the atmosphere into biological usable form, playing a key role in the nitrogen cycle and other chemical cycle. Bacteria and fungi actually create the soil as they slowly break down plants and animal matters, including the leaves in our backyard. This is the picture from NASA, and you can see the bloom of the algae blooms, that's in light blue, and the diversity of bacteria and fungi on agar medium. So now that you know microbes are everywhere, in mushroom production, microbe can be a source of contaminants. Mushroom can become contaminated from many sources during production and processing, including the human harvesting the crops. Bacteria, yeast, and mold cause most of the problem. Growing mushroom from scratch requires a sterile environment and sterile process for repeated success. So you might be wondering, why is this even necessary? Doesn't mushroom grow in a non-sterile environment in a wild? Well, wild mushroom grow in a balanced ecosystem where the environment favors their growth. You cannot create this complex, balanced environment in your home. So if we want to cultivate mushroom predictably and repeatedly, we need to keep it clean. The air is a very likely source of contamination for mushroom grower. Mold spores and other contaminants, although invisible to the naked eyes, are everywhere. For example, a recent study that was done in the U.S. northern Midwest, scientists found 4,076 spores per 3.3 cubic feet. That's double this amount of spores on the outside of the house. Here's a picture of a plate placed on the lab bench. So, how would you, solve the problem of the air as being a source of contaminant? So one way is to use the best option to reduce contaminant in the air is to use a HEPA filter. A HEPA filter is simply a box containing a fan that pushes air from the back through the paper-based filter and then to the front of the unit. As you sit in front of the HEPA filter, filter air is blowing toward you. So if the housing of the HEPA chamber is designed in such a way that the filter air flows in a linear direction toward you, this is called a laminar flow hood. This allows for a clean stream of air in which you do your mycological work, can effectively eliminate airborne contaminant if used correctly. Flow hood or laminar flow hood are quite an investment, whether you buy one or build one yourself. So without a doubt, they are certainly essential to have and make agar work and inoculation so much easier. So you could find one for around $300 to $500 for a new one. For a used one, you could get about $100 to $150. Another option to reduce airborne contaminant is a steel air box. This is essentially a large clear tote with armhole cut up into the side. This way you can clean the inside of the totes with alcohol and slide your hand through the hole to perform the work. A steel air box provides a minimal lab in a box that can be quite effective for hobbyist grower. Here's an example of a portable steel air that you can buy, which is one of this is $99, or you can make your own with a totes, basically. You can also get a laminar flow hood kit for about $399, or as cheap as $56 if you're willing to glue plexiglass or stainless steel sheets on it. If you're near a university like I was, you can really pick them up for free or at a really good discount. As lab close down, they tend to throw it away or donate this to surplus, and you could buy it at a really, really cheap price. Another source of contaminant is the substrate itself. A common reason for the contamination in mushroom cultivation is the incomplete sterilization or improper pasteurization of the substrate before inoculating with a mushroom cultures. The substrate and grain naturally contain competing organisms and dormant spores that need to be reduced or eliminated, otherwise they will try and outcompete your mushroom. Just keep this in mind. Between 10,000 to 100,000 microorganisms live on a single grain of sand, as revealed by a study from researcher in Max Planck University. This means that an individual grain of sand can have twice as many residents as, say, the city of Fairbanks, Alaska. So when you grow something, remember that this is a microbial world. This is a spawn bag that wasn't completely sterilized, so you can see the fungi going out competing the mushroom. Pasteurization is the killing of some but not all organisms in the substrate. Milk, for example, is pasteurized to kill organisms that might harm you, but it's not heated to such a degree that it changes the quality of the milk. The straw, like milk, is not sterilized by the treatment. Eliminating a lot, not all of the organism on the straw is the objective. So pasteurization is needed to be done correctly to minimize the potential for contamination. Straw should be pasteurized at between 150 to 170 Fahrenheit for 1 hour. One way to do this is to use a propane burner and a large 55-gallon drum to create a hot water bath. Sterilization. So supplemented sawdust fruiting blocks and mushroom grain spawn need to be completely sterilized before inoculation. Sterilization of a mushroom substrate is a method of preparing substrate using a combination of steam, time, temperature, and pressure to kill living organism. To sterilize substrate, you need temperature higher than 250 Fahrenheit for at least 2 hours. The time will vary depending on the media you are trying to sterilize. Most substrate grain and agar need a pressure of 15 psi or pressure per square inch in order to completely kill off any potential contaminant. At this pressure, steam reaches a temperature of 121 degrees Celsius or 250 Fahrenheit. The time will vary depending on the media you are trying to sterilize. Liquid sterilize faster and hardwood takes more time. And you can see here, if we have a liquid medium, a quart-sized liquid medium, it will take about 45 minutes at 15 psi. For a quart-size grain or spawn, you need roughly 90 minutes. You need to use a double sterilization method. I'll mention more about this later on. Grain spawn bag, 5 pound, is roughly 120 minutes at 15 psi, and you need to double sterilization method as well. 5 pound sawdust block, you just need 150 minutes at 15 psi. You need to do a double sterilization method because bacterial endospores can resist inactivation from ethanol treatment. They can also survive high temperature for up to 150 Celsius, making gram-positive species heat resistant. Bacterial endospores can also survive at extreme temperatures, almost close to absolute zero. So to reduce this contamination, grain spawn are sterilized for 45 minutes, followed by another 45 minutes 24 hours later to kill heat-resistant bacterial endospores. So the day interval result in the germination of spores. So the spores realize that the heat killed off everything else, so there's no one competing with them for food, so they germinate. And when they start eating the food, you kill them off at 250 Fahrenheit for 45 minutes before they can form spores to survive. This is basically a spawn that was not double sterilized at 250 Fahrenheit. You can see that this is bacterial contamination right here. This section where the mushroom cannot grow into. So can I just boil water or use steam? So boiling water or steam on its own is not hot enough to sterilize a substrate, as the boiling temperature of water at sea level is 212 Fahrenheit or 100 Celsius. Increasing the heat does not increase the temperature of the boiling water any further. So you need to add pressure to raise the temperature to 250 Fahrenheit or above to kill off bacterial and fungal spores. So most mushroom growers will need 15 psi of pressure to increase the temperature of steam to 250 Fahrenheit or 121 Celsius. If you have equipment that can increase the temperature higher than this, sterilization will take less time. Small-scale mushroom growers generally use pressure cooker for sterilization and upgrade to an autoclave or retort or pressure canner as the mushroom business grows. So the all-American brand of pressure cooker are considered one of the best in the class, but it's expensive and it costs around $300. So my Presto pressure cooker has, I've used it for years at home. It hasn't let me down. So if you're a beginning mushroom, if you are a beginning mushroom cultivator, this pressure cooker is one of the best cost-effective. So you can get it used for around $25. So although pressure cookers are simple, they are still serious piece of equipment. If you operate without care, they could turn into a bomb with potential to cause serious injuries. You can see this picture here of someone forgot about their pressure cooker and the pressure got too high, and if there's not a proper vent, it will explode. USA does not endorse using electrical pressure cooker to can things. So in general, you shouldn't use this for mushroom cultivation as well. There's some way around it, but it's not as good as the stovetop one. So grower with large operation use retorts, which are very large pressure cooker, in between the size of a kitchen pressure cooker and a retort or autoclaves, and steam container. Some autoclaves are small enough to fit on a counter. Some are large enough you can walk inside. So an autoclave is basically a chamber surrounded by a jacket. Steam is injected into the outer jacket, which raises the temperature and pressure inside the chamber. This heat under pressure is lethal to kill organism. You can see here, this is a small autoclave machine. This is one that's much bigger, where you can push a whole cart in, and this is a retort cooker basically. So another source of contamination is you. The cultivator themselves are often a huge source of contamination. Your hand and the clothes contain untold number of various contaminants, so consider washing your hand right before doing any lab work. It's also important to use proper sterile techniques, such as paying attention to any contaminant. Just keep in mind that the human body contains 1 trillion microorganisms, outnumbering the cells 10 to 1. This was an old study. Recent studies suggest that this is roughly a 1-to-1 ratio, so there's basically trillions of microbial cells in our body. Another source of contamination is outside organism. For example, there are 3 to 5 million species of mite. This is a conservative estimate. An average person has an average between 1.5 to 2.5 million mite on them. Mites are these little arachnid, a group of insect-like organisms like spider. They cause major problem in mushroom cultivation. You can see there's some mites in this, and that might track and brought in bacteria from the outside, fungal spores from the outside, and created a mess. Nematode is another culprit. For each human, there are 60 billion living nematodes in the soil. So the nematodes population outnumber us. nematode or roundworm that can hitchhike from your body or from anywhere onto plates, onto cultures, onto medium as well. Source of contamination tool. So when dealing with agar or transferring culture, it is common to pass contamination between cultures if you are not properly sterilizing your scalpel. So scalpel can be flame sterilized by holding the scalpel over flame for 30 seconds until it's glowing red. This will help it if you have an alcohol lamp at hand. Alternatively, you can use sterilized toothpick, which I've used single-use sterilized toothpick to transfer cultures and stuff like that. Always sterilize your bench, clean down your bench using sterile technique, which is like a clean bench. So you swab that with Lysol or 10% bleach or 70% ethanol can inactivate a lot of the fungal spores and bacterial spores. Eventually, you might want to maintain a culture collection of different mushroom-producing fungi. So we'll chat about culture maintenance in this lecture. So mushroom-producing fungi will grow on various type of media, which is a nutrient in agar in this case. So first, you want to start with malt extract agar or potato dextrose agar. So you can make them from scratch. So you can take potato, boil them down, mash them, and filter them, and you add agar to that water, and you can make potato dextrose agar that way. Or you could buy mashed potato already, dehydrated mashed potato, add that to water, filter it, and add dextrose, and then you can make your potato dextrose agar. Or you can just buy them. So you could buy potato dextrose agar already made, $17 or $18. You could buy this and you can make about 90 petri dish. You can also buy sterile petri plates as well. You can buy them by sleeves, or you can buy a box, 500 packs, which is much more cost effective. So to do this, you need basically some type of nutrient, water, and agar. So first, you want to sterilize the media, and to do that, you basically pressure cook it or autoclave it at 250 Fahrenheit for 45 minutes. You wait till it's warm to the touch, which you kind of like warm milk, and then you pour the medium onto culture tube or petri plates. When pouring petri plate, practice good general sanitation. So wash your hand, clean the bench top, wipe it down with ethanol, alcohol, things like that. So you want to work under a HEPA filter or a laminar flow hood. This is not absolutely necessary. You could get away with it by working under a Bunsen burner or a alcohol lamp. Works as well. So first, you want to make sure your lab bench is clean. You see this picture right here? There's too many things in here, right? Too many sources of contamination. So spores on this bottle can be blown onto your plate, right? Spores on this box, the spore on this rack. So you don't want your workspace to ever be this busy. You want it to be like this picture here, where you put sterile petri plate, where the sterile air is, right? The sterile air blowing sterile petri plate is okay, and you want to be pouring the plate right around in the middle right here. And you want to lift up the plate just slightly so you could pour the media in. You don't want to just leave the lid open, right? Because spores can fly in. And you can work under, a Bunsen burner or an alcohol lamp. It works just as well. So place your hand between the dish and your body, not between the HEPA filter and the dish. So remember, we just established that. So once the petri dish or the test tube or agar cool enough, they're ready to use. So to transfer it, you open up the lid slightly, you pour the medium in there, and then once the petri dish or the test tube of agar is cool, they are ready to use right away. To transfer a culture from one petri plate to another, use sterile technique. So you take a little piece of the culture, and then you transfer onto new medium, such as malt extract agar or potato dextrose agar. You seal the edges of the plate with strip of paraffin. This is not completely necessary. You can use saran wrap, for example, basically to retain moisture. Store most of the cultures in the refrigerator or better store one in refrigerator and one at room temperature, because some mushroom producing fungi are sensitive to the cold. For example, the straw mushroom cannot be stored in the refrigerator. This is a HEPA filter and a totes. This is basically your laminar flow hood. You can make this relatively cheap. So when you receive a culture or start your own mushroom, you need to make sure that the culture is pure, free of other fungi, bacteria, mites, and contaminate. To do this, so you can't really see your plate and say, hey, you know, this is completely pure, right? You can just, this culture look pretty clean here, and you can assume that it's clear, but you really want to test this by transferring this, right? So do this by cutting out small pieces of colonized agar and transfer each piece to a new agar plate. Repeat at least several times. When all of the colonies grow out, they should look identical to each other. Also be aware of sectoring in a colony on agar plate. We'll talk about a little bit more about sectoring in the next slide. But see this square over here, it's just, you know, you don't want to cut this giant square. You want to take a little bit tip, just this tiny little thing. So this square is just an example of where you're going to target. But when you target in here, just take a little bit, a nick, basically a tiny nick of it. The more media you get, the more chances you can introduce contamination, right? So sectoring is a different in mycelial growth of a colony on agar. It usually appears as a wedge of mycelium that is different in appearance than the rest of the colony. Since the colony grow outward in all direction from its point of origin, in this case, more or less the center of the plate, that's likely where you place a small piece of agar. When you transfer your fungus, a sector looks like a wedge or a piece of pie. It may have different growth rate, color, or texture. The two major classes of mycelium are rhizomorphic, which is strand or ropy, and cottony, which is fluffy. Rhizomorphic mycelia run faster, form more primordia, and yield more mushroom. You will notice the difference when you look at a plate. The change is likely some kind of mutation that naturally arose in the colony, because it could affect productivity of your culture, avoid propagating from a sector point. So this is a clear example of rhizomorphic or strandy ropy growth, and this is fluffy cottony growth right here. So in this first plate right here, agar plate right here, notice this is too big of a piece, right? You don't want to ever transfer this big of a piece of agar, because there's chances you could get contamination. So consider taking a small tip of it, right? And you see this is penicillin, this is a contamination, a fungal contamination, and this is sectoring right here. This section is most likely sectoring or possibly a contamination, but most likely sectoring. So when you transfer this plate, you want to get an edge right here. You want to get an edge, and you want to nick off a tiny piece of this square, and transfer it to a new one. So alternatively, you could take one tiny square here, one tiny square here, one tiny square here, and then one tiny square here, and transfer that all four pieces to a plate, and you can monitor that. So you don't have to go one agar per plate, one transfer per plate. You could do 4 per plate. And this one, you see rhizomorphic growth, cottony growth, right? You don't want to take your culture from anywhere here, right? So again, go for the section that is rhizomorphic and transfer only a tiny bit of this. This culture is all good, right? This one is rhizomorphic right here, and then cottony is forming, right? This one's all rhizomorphic. Here's more example of rhizomorphic and cottony. So changes in cultures is a sign that a strain may have mutated or started to senesce. Your text lists 28 ways to evaluate a strain, but the most important practice is to recognize changes in the culture. So when a mycelium grows old, it's said to be senescing. Senesced mycelium is less vigorous and fertile than young one. So in general, a change from a rhizomorphic to cotton mycelium should be a warning that a strain degeneration has begun. If a culture is predominantly rhizomorphic, then begin to sectors, propagate only from the rhizomorphic sectors. Vary your media so no unwanted selected pressure or exerted on the culture. So what that means is don't use one media. Don't use potato dextrose agar. You could use different concentration of potato dextrose agar. So you use what you need from a culture and then return the rest to storage. So if you have a stock culture, you want to take a little bit piece out and then propagate that and use that. And once that mutate, you go back into your culture collection and start from there, right? The culture collection in the refrigerator kind of slow down the growth, so the fungi is in that state where it's kind of resting and young. So when your culture outside get old, you can always go back into your culture collection and revive that culture. Cultures that have been grown continuously for several years will sooner or later develop substrain that don't resemble the original colony. So if all effort fail, re-isolate new strain for multi-spore germination. This one right here is mostly sectoring now. So if you have to get colony from here, try your best to get a little section from here. This one's beginning to develop some type of senescing, some type of rhizomorphic growth. So you want to go for the section that's much healthier. Same with this one right here, right? You can see the sectoring. Just keep in mind that when you do plate out fungi, you don't have to put one giant agar plug in the middle, right? Make sure you get a section of this, a tiny section of it, because these are microscopic. If you nick a tiny section, it'll grow out just as well, and there's less chance of contamination. For me personally, I like to put 4, one right here, one right here, one right here, and one right here, and watch the growth. So, and I select the best one from there, and I transfer that to make my master plate, basically. So, the first mushroom we're going to grow in this class is oyster. Why? Because oyster mushroom is the easiest and simplest mushroom to grow. See the lab folder, week 2, oyster cultivation, for more photos and illustration for oyster cultivation. Oyster is really easy to grow. You can see I grew oyster in a coffee cup, basically ground coffee from Starbucks. So I went to Starbucks and said, hey, you have some ground coffee, you have a cup, can you put it in a cup for me? And then I took it home and I just grew oyster. I grew oyster in my old molecular biology of yeast book. So you see, I left this outside, and over time, the oyster came out. And this is a different book that I grew oyster on. You can see, completely colonized. Really easy to grow. Very forgiving. So you can grow many species of oyster, all in the genus Pleurotus. So right here you have yellow oyster mushroom, abalone mushroom, pink oyster, king oyster, white oyster, pearl oyster, blue oyster, Indian oyster, and gray oyster. I have grown all of this oyster before. It's pretty easy and pretty fun. So our first oyster that we can grow is, let's grow a pink oyster, Pleurotus dejamor, a beautiful pink oyster. Pleurotus is the genus name, and dejamor is the specific epithet. Together, they make up the species name. No other organism on the planet carry the same name. It's unique to this mushroom. Common names are another matter. They can be many. The pink oyster is known as the flamingo mushroom, the mushroom of love, and many other mushrooms. So private grower and company often come up with a catchy name as a marketing tool for their pink oyster. So when you want to grow a particular mushroom, you need to first determine what it grows on. So how that mushroom make a living. The kind of organic matter a mushroom utilize for nutrient is called a substrate. Fungi are not plant. They do not possess chlorophyll, so they cannot make their own food like plant. Instead, mushroom producing fungi use plant-derived material for their nutrient. Oyster mushroom, for example, utilize cellulose. So our substrate needed to be cellulose based. Cellulose is a linear change of thousands of linked glucose, a type of sugar unit. Cellulose is thought to be the most abundant organic compound on Earth. Organic in this sense simply mean it was or is living, so matter from a living organism. We need to make a technical point here. You would not be able to grow anything on pure cellulose. It has no nitrogen, no phosphorus, no mineral, or other nutrient essential for life. But of course, short of buying pure cellulose anywhere, and if you do, it's quite expensive. So when you say cellulose is our substrate of choice, we are using common sense and realizing that cellulose from natural source contain other compounds necessary to support life. Here, we have student grow oyster, in a Togo cups basically. This is on a toilet paper. So basically, anything that's plant-based or cellulose-based, oyster can basically grow on. So here we have a student using her old genes to grow mushroom. So hopefully she's hoping to end up with natural ribs after that, and some of them did actually have natural rib, but it wasn't this nice. But she was successful in growing mushroom on clothes basically. So what substrate do we use? Let's say wheat grow near your town, and after the grain is harvested, the grower bale up the straw and sells it, or tries to. Wheat straw isn't worth much, and often costs of transporting it makes it too expensive for them to sell basically. So instead they dish it under to increase the soil organic matter, or they let it slowly decompose on the surface. In any case, we got our hand on some wheat straw, which is mostly cellulose. So some people might consider, or some farmers might consider, wheat straw as an agricultural waste product, since the grain has the value and the wheat is kind of useless. So you will see the mush industry often use these kind of waste products that aren't really waste because they contain lots of good nutrient for the soil, but consider waste as a substrate to grow a mushroom. So after your mushroom are harvested, you can return your spent substrate to the soil. In some case, colonized straw can be fed to livestock after mushroom are harvested. So after you're done growing your oyster mushroom on this, don't throw it away, put it in the soil. You might get mushroom popping out, and you might increase the soil nutrient content. So for practical reason of handling the straw, we are going to need to chop it into 4 to 6 inches in piece. Be resourceful, so you can use anything: scissors, your hand, head trimmer, lawnmower, anything to chop up straw basically. This is just a lawnmower that was used to cut up straw in our lab. So it's not absolutely necessary to cut the straw into short pieces. The oyster mushroom don't care if it's short straw or long straw. Cutting it short will increase the efficiency, because eventually you want to grow mushroom for a living. So long stem of straw are more difficult to stuff compactly into a bag or into a box than short pieces. So that's why you want to cut it when you become professional. So we will assume that you successfully cut your straw to 4 to 6 pieces. Next, we need to moisten the straw because mushroom require a lot of water. And 2, we need to eliminate some of the microorganism that's naturally covering the surface of straw. So there's a lot of ways to do this, but eliminating many of the mold and bacteria on the straw is necessary to give the oyster mushroom an edge in the race for nutrient on the straw. So once the straw is wet, the race is on. So a lot of microbes need water to grow. So once you wet it, it's competing with other microbes. So we will soak the straw in hot water, 160 Fahrenheit, for 1 hour to both wet and pasteurize the straw. Pasteurization is the killing of some but not all organisms in the substrate. This is straw that's wet. Once it's wet, you can smell the fermentation that's occurring, the other microorganism that's eating away the straw. So you really want to pasteurize this quickly once you wet it. So to do this, we can buy or you can make a metal tank with a 55-gallon barrel, like I mentioned, or this was used to make for the class. Basically, it's a chicken wire mesh that can fit into this large pot that can be heated over a Bunsen burner. Basically, you chop straw, you can place it in here and heat it up. You can also use steam, basically for the same amount for about an hour or less to pasteurize the straw. This is just basically a 55-gallon barrel, chicken wire mesh. You could dunk it here, and you could light some wood, or you could use some gas, or you can just do this on your stove. So I've actually grown oyster mushroom from home by just boiling the straw on my countertop for an hour and then using that as a substrate. So once you have pasteurized straw, you wait till the straw become an ambient temperature. The straw is ready to receive mushroom spawn. So spawn is the inoculum we use to colonize the straw. Spawn could be the spores, but it's most likely filaments of fungus growing on grain. Remember, straw doesn't have that much nutrient, so this grain actually have extra nutrient that's required for the growth of mushroom. That's why you can grow oyster on like books, for example, on toilet paper, where there's not that much nutrient there. It's because the grain that you use to inoculate the toilet paper or the book has extra nutrient, basically. So by killing many of the bacteria and mold on the straw, our oyster mushroom will grow very quickly. Since this is our first attempt to grow mushroom, we will simply buy spawn. So there's a link in the class handout, but you could buy a bag of spawn and that. So basically, you mix the spawn and the straw together, and then the next step requires to purchase plastic bag with filter patch designed to grow mushroom. These bags are easy to find online, so please go to your search engine and look up mushroom filter bags. You can find them at Amazon, on Amazon, or any store online, eBay, anywhere. So each mushroom bag has filter patch about 2 inches square. The patch allow for gas exchange to keep out the dust, mold, and spores. So as we hand stuff the bag with pasteurized straw, we also sprinkle a little bit of the spawn on top of this. Once the bag is full, we seal the bag with a twist tie or use an impulse sealer that melt the top of the plastic bag together. So this is an impulse sealer, basically. The bag is stuffed. This bag is around 6 pounds. So if you don't want to use any bag, you don't have to. You can basically use a cup like I show you. You can use a trash bag. You can use a 5-gallon bucket with pre-drilled hole. You can use a laundry basket. Like I said, oyster mushroom is very forgiving. I pick up a lot of the 5-gallon bucket. So these are food grade bucket. I picked them up at Panda Express. They have the teriyaki sauce and all the veggie and stuff in this, and at the end of the week, they would throw away a food grade bucket. And you could say, hey, can I take this from you? And they're more than happy to give it to you. And we did a whole class on growing mushrooms sustainably using 5-gallon bucket. So we stuffed the bag with straw and spawn right on a clean table. So, you wipe down the table, you put the straw in there. There's no special room, no special filter that remove the contaminant in the air. No gloves or any sort. You can just use your bare hand if you want to touch it. Or clean hand, just as long as you wash your hand. You can do this with oyster mushroom because oyster mushroom grow really fast and they are aggressive colonizer of the straw. So they can outcompete a lot of the mold that find their way onto the straw. Mold are certainly colonizing straw as well, but most don't grow as fast as the oyster mushroom. So the only thing that was produced using sterile technique in this method of growing oyster is the spawn. So the spawn should be colonized with the oyster mushroom and nothing else. Once you have the bag of spawn or jar of spawn open, you consider that contamination, basically. Contaminate already. So you want to use that up or use that within a day or 2 days and inoculate all your bags, unless you're working under a laminar flow hood. So when I say this, you can think that you can just do a whole mushroom business on just oyster and not invest in a lot of the infrastructure, right? So you just need to buy spawn and straw. That's it, and a 55-gallon barrel. Here you notice some of the mold that came in when I packed the straw, or it wasn't pasteurized all the way, but it's okay. You still have a lot of pins right here that become mushroom. So the inoculate bags is ready for spawn run, which is the colonization of the substrate. We're going to do this at room temperature, so about 2 weeks, 1 to 2 weeks. So depending on how warm it is. So around 80, the oyster grow really well. 75 to 80. You see that the straw has turned white because of the fungus has completely colonized the straw. At this time, the filter patch has allowed air to enter the bag, oxygen in, carbon dioxide out. Remember that all mushroom require oxygen for growth. So in a few more days, we notice tiny mushroom known as pins. So these are little pins. These are cluster of pins that are growing. So once we have this, you could put an X over this to allow more oxygen in, and this will signal this will signal to the mushroom to basically develop this pin into mushroom. So we notice that oyster mushroom grow in a clump. Next, we move the bag to a more humid environment. Remember, you put an X on the bag to avoid it drying out. You can actually remove the whole bag if you have a high humidity environment. That should be fine. So you want to move this to a high humidity environment. You could build your own fruiting chamber, or you could use simple method to increase humidity. Someone built this online, with a little mini plastic greenhouse and the shelf that you could buy, and then a pump with some water to increase humidity. I had a lot of success just using a humidifier. A humidifier in a closet with a timer on to turn it on and off is sufficient to fruit a lot of mushroom. I've also fruit mushroom in totes before, and I did this as a demonstration to the class. Basically, I got a clear totes, put paper towel in here, and I mist the paper towel to get it wet, and then I basically have this little wet container. I put a mushroom block in here, and then I put a lid over with hole cut out, and over time you can see that the mushroom develop. I also just fruit. This is a blue oyster. I fruit outside, so I left this oyster basically in my backyard, and it just fruit. This is another gray oyster that I put in my backyard, and it fruits. This book actually was on my lab bench top, so I left it there, forgot about it, and it started fruiting. So oyster is very forgiving. See, this pink oyster here was left on the lab bench as well too, and you can see that small oyster there was an X that was here, and the oyster came out from it, and it just grew over time. Notice that mushroom need oxygen to fruit. So I was not watching my mushroom bags, and I left this one too long. There's more oxygen in this section, so the mushroom actually bursts out of this section to fruit because it senses that there's more oxygen here. So, and oyster need light, so I left this one in the dark. So this yellow oyster mushroom wasn't that bright, so you need some type of like fluorescent light or some natural light to make the mushroom develop color. So what about scaling production up so that you have many boxes of oyster mushrooms to sell to restaurant in your town on a weekly basis? In that case, you might want to pasteurize a large volume of straw in several drums of hot water. Remember those 5-gallon drums you could get relatively cheap? You want to pasteurize a lot of them and put them into long plastic cylinder bags that you can hang from the ceiling to increase efficiency. So the end of the plastic bag, you can tie a knot on it, and then put a filter, cotton plugs on this, and let air exchange that way. So you can see right here, people put logs, basically plastic bags filled with straw or sawdust, and then at the end they just put cotton filter, cotton ball basically, and then once the mushroom started to pin, they remove the cotton filter, and the mushroom will fruit out from there. It's grow. Also put them in a long plastic bag like this with a cotton ball at the bottom, and let it grow. Once they start growing, they start cutting X's into the bags to let the mushroom fruits. Basically, very low startup cost to grow your own oyster mushroom. So with that, thank you. For the next class, we'll talk about spawn generation technique, starting culture from a spore print, cloning mushroom from fruiting bodies or hyphae, and then proprietary claim, how to start a master spawn jar, the agar characteristic and use, stock culture maintenance and storage, first and secondary spawn generation, grain and sawdust spawn, substrate and inoculation techniques. Make sure to answer the question for your discussion in this week. Thank you.